Hi everybody, my name is Emma Oebel, and I will be talking about iron oxide nanoparticles. So the first question is, what is an iron oxide nanoparticle? So iron oxide is a name for many different natural compounds. There are 16 iron oxides, which include oxides, hydroxides, and oxide hydroxides. Talk about a tongue twister. Some of the most important forms of iron oxide include goethite, which is on the left, and lepidocrosite, which is on the right. And now the next question is, what are nanoparticles? So a nanoparticle is made up of only a couple hundred atoms. It ranges from 1 to 100 nanometers in size and is undetectable to the human eye. So overall, what are iron oxide nanoparticles, or as you will hear me refer to them, IONPs? IONPs are either magmite or magnetite, and they are nanoparticles that are magnetic and are between the sizes of 2 and 20 nanometers, so they show superparamagnetism, which essentially means that they, in and of themselves, don't exhibit a magnetic field unless they're in the presence of a different magnetic field. And this allows them to be more stable. As far as uses go, I'm going to be mainly talking about biomedical uses, and the first one is as a contrast agent. So in some scans, such as CT scans and MRIs, you may need a contrast agent to make veins, organs, and tissues more visible. Tests have shown that not only did IONPs work as a contrast agent, they provided better contrast and lasted longer than the currently approved contrast agents. And another usage is as nanozymes. IONPs have been shown to mimic horseradish peroxidase. The benefits of these nanozymes include easy modulation and high stability despite varying conditions, and they can also be used to enhance nanozyme function as well as as a biosensor. They can also be used in tissue repair and cancer treatment. So hyperthermia is the treatment of cancer by heating the cells in the tumor until they can no longer repair themselves. And IONPs are actually already being used in this way. It is also theorized that they can be used to repair tissues by heating some proteins high enough to join tissues together. This is still very much being tested on. But one thing that has been shown is that while it may not repair tissues, it definitely will protect them from further damage. They can also be used in drug delivery, so superparamagnetic iron oxide nanoparticles can be used to deliver peptides, DNA, chemotherapeutics, and more. And this is a diagram that shows it. So the spion is coated with the drug or the genetic material. It's guided by an external magnetic field to the target cell where the drug dissolves within the cell. And this helps prevent drug waste because you are delivering it exactly where you need it to be. So the next question is, how are IONPs developed and prepared for use? So most IONPs are prepared chemically, about 90% actually, and the chemical process is high yield, low cost, it's efficient, and it gives you a lot of control on many different variations, including size, composition, and shape, which are all important. Physical is the next most common, but it only makes up approximately 8% of preparations. Physical is a elaborate process that requires cutting-edge technology, and so it can be quite expensive. Biological preparations make up approximately 2%, and they are high yield, low cost. High yield, low cost, but they take so long that the cost really doesn't put in that much effect. Another issue that prevents IONPs from being used is that if they're paired in the regular atmosphere, they'll oxidize, which causes issues when they are used. And so in order to combat this, most of the preparations take place in an environment that is filled with nitrogen gas. And then also most IONPs are coated in something like gold or silica to permanently prevent oxidation. So there are some ethical issues when using IONPs. The main one being that There are a lot of side effects, such as lung cancer, skin cancer. It could speed up illnesses or deaths, and it could kill useful microbes. And agriculturally, it could prevent fertilization, and it could prevent plants' ability to restore nitrogen in the roots, which is a big issue. And so what is the issue really? Well, there's just a push for more education on the risks of IONPs. It's still a relatively new biotechnology, and we're still learning what the risks are, but there's a push from a lot of the scientific community to educate patients and those working with IONPs on the risks that they might face. But there's a hope for a genetic test or an environmental study that would tell you your susceptibility to specific risks. Thank you all for listening. I hope you learned about iron oxide nanoparticles, and if you have any questions, feel free to email me. And lastly, these are my works cited. Thank you all for listening,